Karyotyping, in simple words, is the arrangement of chromosomes of an individual in a specific order.
It is an important tool in the branch of cytogenetics as it can detect genetic disorders in unborn babies beforehand and even diagnose diseases like the Down syndrome in adults. This project deals with taking input images of the metaphase cycle of the cell division, which contains highly condensed chromosomes, and then perform chromosome segmentation over these images, and then finally classify these chromosomes to generate the final karyotype. After we obtain the metaphase images of the chromosomes, they are pre-processed in order to be made suitable for segmentation. Pre-processing generally includes refining and enhancing of boundary lines and adjusting the brightness and contrast of the image so that it is most suitable for the segmentation process. Segmentation or disentangling of the chromosome fibers is the first major step in karyotyping. Chromosomes obtained after the metaphase state are highly condensed and fused. A unit-based convolutional neural network is used for this process. The division map is designed to be of the same width and height as that of the input image. A Kaggle-based dataset of 13,000 metaphase images was employed to train the model. The groups of non-overlapping regions are combined and integers are assigned to the class label values. Each chromosome is treated separately and different activation functions and weights are used to get the optimal output.
To analyze the result, we calculated IOU, that is intersection over union, which is a commonly used metric for image segmentation.
It should be as high as possible and preferably 100%. We obtained an IOU of 96.7% for overlapping regions of the chromosomes and 96 to 97% for non-overlapping regions. Since the loss versus epoch graph was of decreasing nature, the idea of checking for overfitting was discarded.
As discussed, the system can be subdivided into four main blocks, starting with image segmentation, which involves disentangling of overlapping chromosomes and separating them in metaphase images, followed by feature extraction, classification of chromosomes as a feature-based mechanism. So some examples of extracted features are size, band pattern descriptors, decomposition of gray level, centromere position, and representation of the banding pattern measured from the microscope images of dividing cells. Next, we come to classification, which resembles the likelihood of class membership and assigning a class to each of the segregated chromosomes. Different types of classifiers like distance and statistical, nearest neighbor, fuzzy, and neural network based techniques have been developed. Among all the classifiers, neural networks are the most popularly used because the model adjusts and learns to different types of databases provided. The final stage includes a graphical representation of the ordered chromosomes. In a standard automatic classification system, the implementation consists of several stages including feature extraction, polarization, pre-processing, classification, and reassignment. Common characteristics extracted include length, area, perimeter, density profile, and contour function. Polarization is performed on the acquired images consisting of uniformly rotated chromosomes to conform with the cytogenist's orientation norm and to offer a consistent variety of features to the classifier. After pre-processing, we implement transfer learning to demonstrate the results using different networks like VGG, ResNet, AlexNet, and DenseNet. We plot the precision versus epoch and observe the accuracy and training times for the different approaches. We use the public dataset which is available with the Biomedical Imaging Lab. It contains 5,474 grayscale images where the classes from 1 to 22 have 238 chromosomes each, while 23, which is the X chromosome, and 24, which is the Y chromosome, have 194 and 44 images respectively. We used 4,370 images for training and 1,104 images for testing. The best accuracy of 94.11% was observed with a precision of 94.51%, recall of 93.86%, and F1 score of 94.18%. This was realized by adding a fully connected layer with 4,096 neurons after the F8 layer. The minimum training time of 2.21 hours is obtained when the FC layer is replaced by a gap layer, which stands for Global Averaging Pooling, which was followed by two of FC ReLU and dropout layers respectively. For the future, we aim to increase the amount of data in the dataset, which should improve the accuracy, and we also want to experiment with data augmentation like reflection, rotation, and stretching. An increase in the convolution size might also reduce the misclassification between chromosomes. This model can also be combined with object detection for a production system that could operate with complete microscope slides.